Only I think two to three people, like only three response I got. Among that, like answers are good. Actually, it's a debatable point. Actually, the either over dominance and incomplete dominance is the exception for Mendel's law or not. Actually, please read the paper. If you read the paper, if you after reading the paper, you will get an idea what answer you wrote, either it's correct or not. After reading the paper, we will discuss. Anybody has previously read that paper? It's a very good paper actually. It's a very short and sweet paper. Anybody has previously saw that paper like Mendel centenary birthday? Actually, the paper was published on the behalf of the centenary birthday of Mendel's law, like Mendel. We also celebrated in IRI with one good conference. All the video lectures are available in YouTube. You can search Mendel Symposium IRA so that you will get all the lectures which are taught in that conference. It was very useful lectures. I hope everyone joined. We'll start the class. Actually, in my point of view, I wrote they are extensions of Mendel's law. Like there are set up. Sorry, in broader heading, I given this also. But incomplete, complete, and over dominance. They're not exactly the exception. Like you can't say that it's non-Mendelian inheritance. We can't say that it is non-Mendelian inheritance. It still follows Mendel's law, but but maybe the phenotypic ratio will change, but the properties of the nature of alleles, what principle was said by the Mendel, like alleles, when they are in heterozygous condition, they will not contaminate each other, and they pass into the pure when they pass into the gametes in their pure form, again they will express the characters. Okay, this is law of segregation. But you can see in case of incomplete dominance, in case of over dominance, we are not violating any principle there. In law of segregation, it's not violated. As usual, the alleles will come in the heterozygous stage and they separate into the gametes in their pure form and they will express again. But maybe the particular allele cannot mask the expression of the recessive allele completely. Universally, you are taking the generally in a broader way. These are not exactly a non-Mendelian inheritance. You can't say that. These incomplete dominance, over dominance, codominance, they are maybe the extensions of the Mendel's law, but we can't say that as a non-Mendelian inheritance. Then, when you're taking the example of cytoplasmic inheritance, you take the example of epigenetics. There is nothing change in the DNA, only there is a change in the modification, the chromatin modification or base modification. There is no change in the actual sequence of the DNA. Then those can be regarded as a non-Mendelian inheritance. But these cannot be regarded as a non-Mendelian inheritance. They may be extensions of, not exceptions, like extensions of Mendel's law. But in many books, it is wrote exception. But as per that question, have to answer in such a way that first you should tell the laws. As like Satyam wrote the very beautiful answer. First, you have to state the two laws. What law of segregation is saying, what laws of independent assortment is saying, so that then you have to give the example of incomplete dominance and the over dominance. Then you have to show that where up to what stage these two conditions following the Mendel's law, at what stage they are, say, the why the confusion arises, at what stage. At phenotypic ratios, the confusion arises, but while the segregation and independent assortment is the same, it follows the same Mendel's laws. Okay, that's why you have to write the answer in such a way. First, you have to explain the Mendel's laws. Then you have to write what are the condition in incomplete dominance and complete over dominance. Then the similarity, like where the law holds good, where the law confuses. Like why you saying that it's extension or exception? Answer should be in that way. Then you can fetch a 10 out of 10 marks. With that, we'll start a new section of the class, like a new part of the class. Anybody have doubt in that? I'm audible or not?
Sir, also explain that linkage, sir, linkage.
Yeah, exactly. You already wrote a very nice answer, but I will explain
Thank
again.
you, sir.
See, as you said, like I read your answer. It's very good. Like for example, there are I yesterday in detail like I told that there are five linkage groups. Only two traits are in the same linkage group. See, only two traits, for example, like for stem length. Yeah. See here, this one and this one is in the same linkage group, stem length and pod form. And another one is
Shade
5
shape
and 5.
and
Yeah,
pod colors.
seed shape and pot color. But the Mendel was published paper, na, he didn't take this combination. Okay, that's why maybe he didn't got linkage. Maybe that's why Mendel is lucky. Even though these traits are together, same linkage group, but they are far apart. Okay, they may not. Luckily, they not show any tight linkage because of the distance is very high in between them. This may be the two reasons. One, the combination he not chosen for the while submitting his paper, and also he now maybe those genes are the very far apart in the same linkage group, so that there is no tight linkage. That's why he did get any deviation from the normal dihybrid cross. We will discuss how linkage affects the dihybrid cross ratios in the next coming class, today's class. Clear? All of you clear with this to the full answer?
Yes, sir.
Sir, one doubt, sir.
Yeah, yeah, yeah.
Sir, not regarding this, sir. Sir, as I was in exam today, I can't able to send the answer, sir.
Okay,
But
okay, no problem.
one doubt is there for me, sir.
Yeah, yeah.
Uh, in
Clear.
case of over dominance,
Yeah.
is environment only plays the part, or the metabolic block or reaction pathways is also there, also?
Yeah, many things, molecular, biochemical, everything plays important role when it comes to our dominance. But it's not exactly the property of the alleles, but maybe the property of the heterozygous condition.
Okay, sir. Okay,
Yeah.
sir.
It's not actually because of the allelic nature, but when they come together in a heterozygous condition, they surpasses the both the parents. Due to the heterozygosity. When you are studying the heterosis and their genetic hypothesis, you will come to know like what exactly the over dominance. I think many of you heard over dominance hypothesis of while explaining the heterosis phenomenon. We will
Yes, sir.
explain
Yes, sir.
detail there. Okay. We'll start today's class. If any doubts, no doubts is there. I hope everyone clear with that extensions and exceptions, assumptions of Mendel laws. Actually, I'm going according to the syllabus. Many of you think that I'm going in a random way. It's not like that. I was going according to the syllabus. You're able to see my screen, or you're only seeing my presentation?
Presentation, sir.
Okay.
Screen, it's of entire screen, sir, actually.
Like folders are visible.
No, sir. Slide share is only there, sir. PowerPoint
Without
is
slideshow,
open.
sir. PowerPoint.
Yes, sir. PowerPoint is
Power.
open.
Okay. Actually, I opened the folders. Now syllabus is visible.
Yes, sir.
Yeah. Actually, I'm going according to the syllabus. Actually, it's a ARS syllabus. SR Bennett syllabus. Like as October 8th, recent, like earlier it was for PhD, but now they made it for MSc on the basis of MSc, like minimum education quality respect to the, it's a revised actually. It's a revised. Actually, they again made MSc. It's clear that okay, if anybody have confusion, like either PhD is compulsory or it's already made MSc as a compulsory. Actually, many were asking like preparing for SRF and preparing for this one is different or it's same because syllabus is copy pasted. What are the syllabus here there? They just copy pasted. Even the mistakes were there in this spellings that also there in the SRF syllabus. You can clearly observe somewhere. Okay, that's what lazy. Okay, see, like I have covered the Mendelian inheritance. Now I will cover cell structure and linkage, how we have to estimate and now. And epistaticus we have discussed, gene concept I have discussed, allelism, how to test the allelism, complementation test. I told to read because it's not that much in detail they learned us. Fine structure of the gene, up to here we discussed. Now from extra chromosomal inheritance also we discussed like maternal inheritance and cytoplasmic maternal effect and cytoplasmic inheritance and this, and we will discuss again extra chromosomal inheritance while I am discussing the male sterility systems. Then little bit I will discuss these things also may be possible today. And genetic code, gene enzyme relationship, replication, transcription, translation. We had to. I will not cover this one because if I tell if more detailed information available in the YouTube, any lecture you watch any molecular biology, those who are very good with Hindi, please refer which is that Academy for Life Sciences, Shomush Biology. Follow his lectures on replication, genetic code, repair, translation. Excellent teacher actually. Even I also followed him. Even I think that much detail is enough. Like you have to need either for exam. Don't worry. For exam for SRF and ASRBNet, nothing will come from these things. Only 2-3 bits, maybe maximum 2 to 3. That is very easy questions will come. Like for example, which is the enzyme is responsible for the DNA replication out of polymerase 1, 2, 3, which one is the very important? Polymerase 3 is the very important. Which one is for mRNA production? RNA polymerase 2. Which is for tRNA production? RNA polymerase 3. Like this, very basic questions will come in both plant breeding and also like in ARS, ASRB plant breeding or in genetic SRF genetics plant breeding. No need to read in detail about it, about molecular genetics. But although it is not needed, but you have to read because everything is revolves around it. And please refer that Shomush biology lectures for this one. And because this is not that much conceptual with animation, these things. Now, it's like not only show much biology, you watch any videos with animation so that you will get a more in detail idea about replication, translation, transcription. Rest all the things I will deal in detail, like plant breeding. These are things nowhere you can't find any lectures in anywhere. Okay? That's the one of the lacuna in the science of plant breeding actually. Economic botany I will deal. Never find any lectures about these things. I will deal with this. And genome organization I will deal with because you can't find anywhere these resources. Quantity genetics are the same. Maybe you can refer Vikas Mangal's videos, but I will deal in detail. And this thing I will deal very in detail eh, for the marker to marker restricted genomic selections. Sorry, this also I will do because I done it. I will explain. And these things I will do in detail, and these things also I will go in detail. Statistics also I will deal in detail. But I will not detail in detail. I will not go enough like in detail about genetic code, gene enzyme relationship, replication, transcription. These are the beheading things you should remember. Okay? Then, as for the chromosomal, we will discuss a new topic: chromosomal basis of inheritance. I'm audible? Like, please respond.
Yes, sir. Audible.
Chromosomal basis of inheritance. As we look into the Mendel's laws, it's clearly in detail, like how the traits were transferring and segregating from one generation to the other generation. But what is the basis of that inheritance? Who carrying? We said that the factor is transferring. But who carrying? Some vehicle should be there to transfer this factor from parent to progenies. But in 1806, there was developments inside in 1880s. There were a great development in the field of cytogenetics. Okay. I think in 1888, the Waldeyer named in 1886, I think Strasburger identified the chromosome. He was discovered the chromosome. In Waldeyer named it as a chromosome. First, he just identified. Then Waldeyer has named it as a chromosome. Then, after identification and there is development in the chromosome and the structure, and later, the one remarkable development happened is chromosomal basis of inheritance. Can anyone tell who given this term? Like who is given this hypothesis? Chromosomal
Saturn and
basis of
Bovary.
inheritance. Exactly. At the which year?
1902, sir.
Yeah, exactly. 1902. He told that what is chromosomal basis of inheritance means. See the behavior of chromosome. Before that, understanding the chromosomal basis of inheritance, you should know the how cell cycle, what is mitosis and meiosis. You can see that there are cell cycle, there was G1 phase, S phase, G2 phase, and we have M phase
Excuse
called
me,
as
sir.
either hello? Anybody have
Yes,
doubt?
the presentation is not visible, sir.
Sorry. Should I tell them? Sorry. Uh, sorry. Thank you for informing. Now it's visible.
Yes, sir. Visible.
Yes, sir. Yes.
Thank you for informing. Then you know that chromosomal basis of inheritance was given by Sutton and Boveri. Actually, like factors were there. Mendel has told that factors governing for the trait, they are transferring from parent to progenies. But someone needs to be there to transfer this factor from parent to progenies. And Mendel also said that they are particulate in nature. particulate in nature. But earlier it's believed as a fluid in nature. The genes are the factor which is responsible for the phenotype development is fluid in nature earlier. Like the pre-Mendelian scientists had believed in that theory. But Mendel said that it's a particulate nature and having an alternative forms. If if the factor is in particulate nature, somewhere it has to be present in the cell, or somewhere it has to be present in the body, in the plant, that has transferring from parent to progenies. That evidence came by the discovery of the chromosomes. Like in 1887, like 18, if I- 1888, Waldeyer named, and 1886, I think, the Strasburger was discovered the chromosomes. After that, the development in the cytogenetics was happened. That is called as a chromosomal basis of inheritance. You can look at this. There is was a cell. There was a cell. Then there is a nucleus. There is a development in the cytogenetics and cell, like cell biology also. With that development, this theory came to existence. Nucleus, in that there will be a chromosomes, in that there will be a DNA. Then we are at the stage of at nucleotide level, we are playing at nucleotide level. But at the time of 1880s, still we are in the stage of studying the cell and the cell organelles, chromosomes. Okay, at that time you have to think now. Then there is a development in the understanding the division of the cells. You have to, and if you want to understand the chromosomal basis of inheritance, we need to understand how the cell will divide and how the chromosomes will behave, or the properties of the chromosomes throughout the process of the cell cycle. We should know that how the chromosomal structure will change and how it passes through the different daughter cells during the cell division. We should know. Then only we can able to understand the chromosomal basis of inheritance. I think many of you know that I am going very at a very high speed. As we know that cell cycle having the G1 stage, S stage, G2 stage. But G1 stage, anyone tell what will happen? It's a preparatory stage. Okay, like what are the requirement for the DNA replication? It is synthesized, like it will synthesize RNAs, it will synthesize enzymes, it will synthesize proteins, so that it can able to serve as a food for the replication process. Here, our in S phase, chromosomes will replicate, or DNA will replicate. Then again at G2 phase, then it will enter into the mitosis, or if it is in the germ phase, it will enter into the meiosis. As we all know that prophase, metaphase, prophase, metaphase, anaphase, telophase. And I'm not knowing in detail like what happens in the different stages. It's a very basic thing. You should know that what will happen. Last year in SRF, the question was asked. I already discussed this membrane. Like what are the cell membrane will be there, a nuclear membrane or cell membrane, cell wall and nuclear membrane. This will completely disintegrate at the prophase 1, at the prophase stage, either in the meiosis, normally asked in meiosis, statement 1, and again some stages longest or shortest, anaphase is the shortest phase you think. Like either both statements are true, both statements are false, one statement is true, one statement is false, like that the questions were arised. Very basic questions will arise. Okay, you should be very thorough with the basics. Here at prophase, cell wall is not completely degrading. It is completely degraded metaphase stage actually. You should know that. Then, which is the shortest phase, which is the longest phase, and where exactly the chromosome will double at what stage. And you should know checkpoints, where the particular checkpoints are there. Can anybody tell at what stage the checkpoints will be there?
Sir, in S phase, at the end of S phase, whether there is a complete replication of DNA, there is a checkpoint, I think so.
Yeah, Yeah, exactly. Actually, checkpoint will be there after, like during starting of the M phase, and after the G1 phase there will be a checkpoint. Please go through that. Uh, what is the checkpoints actually? What are these checkpoints?
Since they regulate the cell cycle, I think so.
Yeah, exactly. There will be two terms. Like, please remember, there were a scientist associated with. I also forgotten actually. There are three Nobel laureates who actually developed the cell cycle regulation. Okay, please. Actually, I'm not getting their names. It's very difficult to remember their scientist names. There are three Nobel laureates. You should remember that things actually. It's very important. There are three Nobel laureates. There are three Nobel laureates who are first who developed cycling. You type in Google. Who developed cycling? Who developed a checkpoint concept and CMD mutations. I remember this, but I can't able to remember the scientist names. This anybody can search. I'm just continuing. If you are searching, if you find, please answer. Who developed the cycling concept? Who developed the checkpoint concept? Who developed the CMD mutations? Okay. You should remember these three scientist names and what are the checkpoints are present across the different cell cycle. And you should also remember what is the role of the phosphokinase enzyme. Like there will be a phosphorylation or dephosphorylation of the cyclin at each checkpoint level. You should know if phosphorylation happens, what will happen. If dephosphorylation will happen, what will happen. Actually, these are the mugging of things you should read and you should remember and you should forget after the exam. Okay, I will not go in detail. Please read these things. Then we'll come to chromosomal basis of inheritance. You can see that when there is a cell, Priyanka, for example, there is a chromosome. You know how it will going to divide into the, if it is a mitosis, means if it is a 2n, you get 2n, 2n. So simple mitosis is, it's an equational division. But in case of meiosis, what will happen? You will get four daughter cells and it's a reductional division. You get haploids. What is exactly haploid means? Can anyone tell what is exactly what will happen?
Only one set of genome is present.
Yeah, exactly. There is only one set of the genome is present. Here they will get the chromatid will first the chromosome homologous chromosomes divide, then again the chromatids will divide, then again they become till of they become again daughter cells. Means how the genes will segregate? T and T will be there. First, the they will segregate, homologous chromosomes will segregate apart, then A will segregate, A will segregate. Here it's very clear. Sorry. You can look at first in leptotin what will happen? They will come to near, and in zygotin what will happen? There will be formation of bivalents. There will be formation of bivalents. Then there will be a crossing over. then there is a diplotin, then there will be diakinesis where your chiasma will end. In the same way, your alleles will segregate. Okay. First, for example, you take this is a homo like capital A, and this is small a. How the chromosome will segregate, the alleles will also segregate in the same pattern. Means the behavior of the segregation of allele is same as the segregation of chromosomes in the cells division. At what stage it's similar? Like at what stage the segregation of the chromosomes to the different poles happens? Sorry.
Anaphase.
Yeah, anaphase. Okay. At anaphase stage, we can say that this is the basis for the this is like experimental basis or physical basis for the your segregation of the particular alleles. Okay. I think many of you understood it's a very easy topic. Any doubts regarding this? What is the physical basis for the Mendelian laws of segregation? Anaphase 1. But there is another thing. If it is not no crossing over, it will happen in the anaphase 1. If there is any crossing over will happen between these homologous chromosomes. Crossing over happens in homologous chromosomes or non-homologous chromosomes.
Homologous
I will
novel.
come. Yeah, homologous chromosomes. If there is no crossing over like this, if they separate into different poles and different gametes, they will segregate at anaphase 1. If there is any crossing over, the genes will segregate at anaphase 2. The alleles of a particular genes will segregate at anaphase 2. You should remember, when there is a crossing over, segregation will happen in anaphase 2. When there is no crossing over between the gene and the centromere, the segregation will happen in anaphase 1. If then next, I think here I will be very clear. So you can check how the traits. Here you can see the Mendel's law. Here you can see. For example, this is the recessive allele. This is the dominant allele. They present in the homologous chromosomes. You can see at the metaphase, they will be at the metacentric plate. When they move to in the anaphase phase, they will move to the different poles. You can see that the small y will coming into the different daughter cell and capital Y coming into the different daughter cells. Then again the meiosis 2, there will be a separate 4 haploid gametes. You can see. This is how you can see the segregation of chromosomes along with the segregation of the alleles is exactly the same. This we can regard as this is actually the experimental evidence for the Mendel's law. There are any other experimental evidence other than this? Can anyone tell? It's clearly visible. Na? I think no need to explain also. It is like explanatory, the diagram. Anybody have doubt in this? How the allele will segregate according to the segregation of the chromosomes in the anaphase? And anybody have doubt?
Sir, I have a doubt
Yeah,
regarding
yeah,
that cell membrane breaking
yeah,
after the prophase.
yeah,
Sir,
yeah.
is it cell membrane or nuclear membrane, sir?
Actually,
Because
it's a nuclear membrane and both the cell wall actually. But in book it is given cell nuclear membrane. Actually. Sorry, nuclear membrane.
yes, sir, because the cell divides only during the telophase, na,
Yeah,
sir.
yeah. Actually, it's a nuclear membrane. Actually,
Hmm.
it's a nuclear membrane.
Yes, sir.
Okay. Yeah, it's a nuclear membrane. Once you check,
So,
actually, I need to check. I need to check. Actually, I have confusion in that. Okay, I will confirm tomorrow. Okay,
Okay, sir.
either nuclear membrane or cell membrane. I have confusion. I think both will disintegrate.
So
Because you see,
because if the cell membrane disintegrates, the cell contents
yeah,
will flow out, na, sir. They will come into the intercellular
no,
spaces.
actually, when there is a new formation, new cells, the first forming component of the is middle lamella. That statement also
Okay.
there. Wait, I will confirm now only. Storage.
I have seen somewhere, sir, that during telophase, the middle cell wall plate will form without
Yeah, exactly.
breaking the original cell membrane.
And sir,
Then,
another thing, sir, this karyokinesis, it is the first step in cell division.
yeah, exactly. It is there in this one.
So, sir, this nuclear membrane is appropriate, I think.
Yeah, but I have to check. Mm, yeah, exactly. Yeah, in metaphase, there will be a disintegration of. Yeah, exactly. The nuclear and disintegrate in the metaphase. Thank you for correcting me. Let's breakdown of nuclear and wall of appearance of spindle fiber. Exactly. Sorry for the. Actually, it's not a cell membrane; it's a nuclear membrane. Thank you for correcting. Exactly. It's only the nuclear membrane will disintegrate at the metaphase stage. Okay. Now everyone clear what exactly he is saying? Actually, he is correct regarding the disintegration of the particular membranes. It's a nuclear membrane. It's not a cell membrane. It's not cell wall also. Okay. Anybody have doubt that what is nuclear membrane, what is cellular membrane? You can able to ask. Like you can ask. Okay, no doubts.
No,
Yeah, this is the same thing again. Independent assortment. Yeah, I think many of you know that what is sex determination. This is the one of the evidence for the again Mendelian basis of inheritance sex determination. It means that genes present on the chromosome. It proves that gene present on the
Sir,
chromosomes.
sir, sorry for interrupting, sir. I have
Yeah,
doubt
no.
in
You should
previous
always
slides.
interpret this one.
I have a doubt in no, sir. The segregation. Yes, sir. This slide, sir.
Yeah,
As you
yeah,
say, sir, the crossing over will happen. But if
yeah,
there is a crossing over, the gametes will segregate in meiosis too, sir. But
yeah,
I
exactly.
think there will be an exemption if there is a second division restitution, sir. In case of such kind of restitution in developing polyploids, the gametes can segregate. We
No.
can
That
say
actually that is another thing. While explaining the polyploids, we'll come to that point. Actually, you are saying the
yes.
correct. There will be a double reduction will happen when there is a tetraploid conditions like tetraploids. Like when there is a auto tetraploids, when there is an auto tetraploid, there is a special condition that is called as double reduction. I will explain in detail when it comes to polyploidy. Actually, there is a possibility of getting, for example, there is a simplex condition. Means we think that there is no possibility of getting capital A allele, but still we will get a capital A gene, like this one, gamete. How it possible? Because we having only a single out of autotetraploid, we have only single A dominant allele, and other three were recessive allele. This is called a simplex. But although due to double reduction, we will get capital A. There is a different phenomenon called as a double reduction phenomenon. So while explaining the like autopolyploids, I will explain this concept. Okay. Now you clear what is this concept is?
Yes, sir. Sir.
Yeah. Then it is like how we can explain the law of independent assortment on the basis of cell division. Okay. This is another example evidence that the genes are present on the chromosome. As the chromosomal basis of inheritance also says that our genes, these genes are the factors they present on the chromosome. They inherit according to how the chromosome will segregate in the cell division. The alleles of the particular genes also segregate according to that. This is actually a very simple. You just remember genes, the segregation of the genes is similar to that of the segregation of the chromosomes in the cell division. Then, when it is come to sex determination, we know that in the chromosome we have two types of chromosomes. One is allosomes and one is autosomes. What is allosomes and what is autosomes? Can anyone?
The allosomes are sex chromosomes and autosomes are the normal chromosomes.
Yeah, exactly. Then,
And allosomes are different structured chromosomes. Means they're structurally different, but
see,
autosomes have homolog.
if I ask you for 5 marks, what you will explain? In terms of the pattern of inheritance,
They
can you
they
explain
won't follow
allosomes?
the same
Exactly,
inheritance patterns, chromosomal inheritance.
exactly. The allosomes will follow which type of inheritance, like which pattern of inheritance?
Sex limited patterns, right?
Can you repeat again? See, these are
Sex
the
limit,
basic things.
sex.
This that is a different concept. I'm asking that how allosomes are different from autosomes. If the genes present on the allosomal chromosomes, what pattern of inheritance they will show?
The
If the genes
sex linked
present on the
inheritance.
autosomes, yeah, six linked inheritance. What pattern they will follow? Crisscross. Yeah, exactly. Crisscross inheritance. Then then I come to that. Somebody is telling like sex limited, sex linked, sex influenced. Can you explain what the terms? How it is different from this concept? Either it was sex limited and sex influenced. They are the genes which are involved in sex limited and influenced. They are present on allosomes or autosomes. No question. Can
Or
anyone tell?
Autosomes.
Exactly right answer. It's they are present in the autosome. Sometimes maybe they will present on allosome. That can also a possible, but the genes they are in the autosomal chromosomes, but they show a sex limited or sex influenced characters. Okay, even though they are not present on the sex chromosomes, but they show the characters based on the sex of the particular individual. Okay, understood what I mean to say?
Yes.
Now you got now you got an idea that what is sex linked, influenced, and limited. Even the genes, sex linked means the genes present on the sex chromosomes. Genes which present on sex chromosomes. They are called as sex linked inheritance, the trait which following sex linked inheritance. But the genes present on autosomes, but the expression is limited or influenced by the sex of an organism. Okay, those are called as sex limited traits and influenced traits. What are the example for sex limited traits in plants?
And the development
Exactly, so same. And the development is limited to male,
is
but the
in
genes may be
development.
present on, yeah, even the tapetum development genes and the autosomes, but it is limited to male. there will be a great example, the great sense of thing. Okay. Normally, we if you'll remember the example which present on the books. Okay, this is really a good thing. Then sex influencer traits. You can say any example, either in plants or humans, animals. There is a first example, sex influencer traits in sheep.
Baldness,
Okay.
baldness in humans,
Yeah,
and
that one.
horns in sheep also.
Yeah, harmlessness in sheep. Okay. This is the things. These are the things you need to be very clear. Okay. Where the regarding the pattern of inheritance. You told that the allosomes will follow crisscross inheritance. Why the genes present on the allosomes, allosomal chromosomes like X and Y chromosomes, will show crisscross inheritance? Is there any particular reason for that? Today I am very little bit fast because I need to cover so many things in this. Can anyone tell why the genes present on the allosomes will follow crisscross inheritance?
Because they don't have their counterparts.
Exactly right. See, you are seeing here. There is unbalance in the, for example, in the female, you take in case of humans, here XX is there. In the male, only XY is there, no X, another X chromosome. Then definitely the gene present on the X chromosome, the genes present on Y chromosome can never be present in the female. Then it's obviously show the different inheritance pattern than the your Mendelian pattern of inheritance. In Mendelian pattern, both are same. Here A chromosome will be there, here also same will be there. But here the absence of the particular genes in the counterpart sex. Okay, that's why it is show crisscross inheritance. I think everyone got to this point. Any doubts in this? Why
No,
the
sir.
particular yeah. Yeah. See, there are several examples of sex determination. There are in different organisms, there is a different terms, like the different basis of sex mechanisms. You can see in the humans, we already know XX, the homozygotic in females, but heterozygotes in the males. But in the case of Drosophila, like say this is grasshopper, there will be a X, there is no O, like XO and XX mechanism. And in the case of hen, there is a ZZ and ZW. But here you can see the female is heterozygous image. But there is a special type in the honeybee where the haploids are the males, where diploids are were females. Why it is happens? Anyone tell? Why is this special?
Parthenogenesis happens.
Yeah, exactly. It's because of haploid male is developed through parthenogenesis. This is why you should only remember in the exam point of what is allosome, what is autosome. Based on the genes present on the allosome, the genes present on the autosome, we can classify the sex, the trait limited to the sex, like sex influenced, sex limited, and linked. Okay. Then you have to remember which type of the mechanisms will determine the sex of an individual. Like here, the homozygous allosome XX will define the female and XY will define the male. Here in Drosophila, there is a different mechanism. In hen, there will be different mechanism. You should mug up. Then there will be separate mechanism in the honeybee where the haploids are produced due to parthenogenesis. That's why the haploids are the males, where the diploids are females. There again is different classification based on the feeding of the royal gel, like what do we call? Uh, then the queen will
Royal
be there
jelly.
and the work. Yeah, royal jelly. Then there will be a workers. Okay? Should know these points and how this exactly works. Then the mapping. Actually, cover and I had to go with analysis. Today, actually, I thought to start linkage and recombination chapter. Tomorrow we'll discuss. Then this is the gene mapping. We will discuss this one. Actually, I thought to start. The gene mapping is another evidence for the Mendelian inheritance pattern. Okay, in 1932, the first genetic map created. Actually, it is not the genetic map. Just for a symbolic purpose, I mentioned. First genetic map statement as created in 1930 something. Then it's clearly exhibited. The gene present on the chromosome, we can able to map it. Okay. Who worked in Mage regarding the crossing over? Who actually discovered that the crossing over, the mechanism of crossing over, there is a let's say exchange of the chromosomal part between the homologous chromosomes. Who is the scientist? In Drosophila, there is a separate scientist, and in Mage, there is a separate scientist. Can anyone? He said that the crossing over is the mechanism where the part of one homologous chromosome is shifting to another counterpart of homologous chromosome. Exact evidence with knobbed chromosome experiment she given.
Barbara McClintock.
And
Storm.
another one,
God storm.
Stern in Drosophila. Craigton something is over there. Clintock and Craigton, like Craigton. Okay. Remember that experiment. Which were the chromosomes he studied? You should know. In Stern experiment, which are the traits he used? Attached X chromosome, okay, bar I trait, and apicot. Sorry, that one I trait. Bar I trait, attached X chromosome. Another one is like apicot I something will come. Red apicot, red and white. You should remember all the three things. And in here, knobbed chromosome. Chromosome number 9, there will be the presence of knob. Based on that, based on this morphological evidence, cytological evidence, they proved that there is an exchange of segments between the homologous chromosomes in the process of crossing over. You should remember that experiments. Okay. Then the another evidence, the ultimate evidence is the proof the genetic material, that is like fine structure of the genes and the exact structure of the DNA. Okay, this is how up to this the Mendel's laws, gene concept, and chromosomal basis of inheritance, and all the sex limited characters and these things. This is the basic general genetics everyone should know. Then it's a molecular genetics. From here I will start a molecular genetics. We discussed what is gene, and we know that now where it is present on the chromosomes. Then we should know who carry it.
Gametes.
Who carry it? Can you again tell? Who carry it?
Gametes.
Yeah, gametes. Okay. I'm talking at the cellular level. The gamete having the cell.
Chromosomes.
Yeah, chromosomes. On the chromosomes, the DNA. Okay. Now we are in chromosomes, there will be DNA packed. In that DNA, our DNA sequence was there. We discussed what is the operational definition of gene. You only told. It's actual sequence of the DNA from 5' end to 3' end. Okay, it includes promoters, terminators, coding sequences, introns, exons, coding for a functional product. Means the ultimate place where the gene is present now at in the DNA. Okay, this is actually the ultimate evidence. Okay, then who actually discovered the DNA? Is Frederick Mitchell the first time in the 1868. Then again, Altman actually isolated first time in 1889. Then Griffith experiment He just say that something is there, it transferring from one to other. Then this experiment, Avery, McLeod, and McCarthy, this experiment actually proven that this DNA is a genetic material. Yeah, actually, gene mapping. Actually, I will be discussing in the next coming slides. Okay, I will come to that in detail. Then, in the last year, in 2024, the question was asked. Actually, there were a question on the molecular name of, like the chemical name of the cytosine, or cytosine or thymine, I think. Actually, many of you forgot on this. We never read this. Okay.
Sir, it's
Adenine
very
means,
difficult to remember, sir.
yeah, actually, it's very difficult, but it asked in the exam. Okay.
Frequently, I'm getting forgot.
Yeah, actually, even I also forgot. That's why normally I will not add any contents in the slides. See, I will remember and I will come. Actually, I will read and I will keep it in script notes, and I will come. But those things are which very difficult to remember. I will write. Actually, I will write in the slide. Okay, it is actually the logic why I written this. Actually, I know it's very difficult, but for your normally we neglect it because of difficulty in remembering. We neglect it. Actually, we should not neglect it. It is a matter of 5 marks, not of 4 marks. Normally, we'll attempt. Okay, we'll lose -1. Along with our 4 marks, your minus will also go. Means 5 marks we'll going to last. Because simple this. If you remember these things, you get 5 marks. But many this time they asked in very this time question paper was very tough. Wherever we neglect, he pointed out there only and he asked the question. Actually, this was a trick he played actually. Wherever we not remember, like we'll neglect it, he picked those concepts and he directly pasted in the exam. He asked cytosine and thymine, I think. He asked exact names. 2,4 dioxy, 5 methyl pyridine. Okay. You can remember adenine, 6 amino purine, nothing is there. Because adenine we know that purine and guanine we know that purine. Only thing you should remember these groups, amino group, amino 6 oxy, and cytosine, thymine, uracil. They are pyrimidine. But you should remember, and thymine and uracil only difference is
Methyl
Methyl
group.
group.
yeah, yeah. That's why. See, if they given uracil and thymine, you should know when there is a methyl, you should directly uh, making like you should eliminate the option uracil, because in uracil there is no methyl group. And if its amino group is there, amino group is specifically for your cytosine. You should remember that amino group is specifically for cytosine in case of pyrimidine. But in case of purine, it's present in both, in adenine and guanine. But you should remember among the pyrimidine, methyl group is only specific to thymine, and amino group is only specific to cytosine, and there is nothing in the uracil. But among the purine, among the purine, there is no oxy. You should remember like this way. If you directly remember the adenine, 6-amino purine, guanine, 2-amino, it will not work actually. You should remember in that way that which is not present in which. Okay. First, you classify 3 pyrimidines, 2 purines. Among the pyrimidines, what is the difference? They are all pyrimidines, but why they are different? Among uracil thymine, there is absence of methyl group. If there is a presence of methyl here also, they are exactly one another the same. Then in cytosine, there is a presence of amino group, but no other pyrimidine having amino group. Like that, we should remember by making it as a conceptual. Okay? And all the pictures and the information I am looking, actually, I am reading another book, Robert J. Booker. It's a very beautiful book. Even a 10th standard student can able to understand the genetics. He wrote in such a way, in a simple way, with all animations. See, all the things I took from this book. You refer if you are. I will share the PDF. If you have time, you refer this. Okay. Then, what is central dogma? They will not go what is 5 prime to 3 prime, anti-parallel nature, how many bonds present between adenine and thymine, guanine and cytosine. All those things will not going to deal because it's everyone know. There is a GC, there will be 3 bonds, there will be AT, there will be 2 hydrogen bonds, there is a hydrophobic bonds. How their stacking will happen, which is the main structure, which is the bonds holding these molecules together. Can anyone tell what are the bonds? One is hydrogen,
Phosphodiester.
yeah,
Phosphorus,
phosphodiester bonds, hydrogen bonds, okay,
glycosidic bonds,
glycosidic bonds. And if you're adding a one molecule of nucleotide to the growing chain, how many water molecule will release? That thing you should know. How many water molecule will release?
can you
See, if
repeat the
you
question, sir?
can you repeat?
Sir, repeat the question, sir.
Yeah, I will. For example, you are adding a nucleotide to the growing chain of DNA. How many water molecule will release? Okay, let it be. How many pyrophosphate will release? Your nucleotide
One.
having how many phosphates? Your nucleotide having how many phosphates? First question. Your
Please.
nucleotide has having.
Sir,
Can
I
you
think
repeat?
only one, sir.
No, I'm asking nucleotide. If you are adding one nucleotide, like normally in which form you will add a nucleotide? Can anyone tell the full form? DNTPs. Who are done PCR in their MSc? They know this better. What is DNTP?
Deoxyadenosine triphosphate, like
No,
that.
no, no, okay. Triphosphate. In the name itself, it's there. Triphosphate. Okay. Normally, we'll add DNTP to the growing chain in the PCR thing. Either all the three phosphorus will embed it into the nuclear chain or not.
No, sir.
Yeah. Which phosphate will going to embed it into the nuclear, like it will going to add to the growing nuclear chain?
Alpha phosphate.
Yeah, exactly. Other two phosphate releases?
Pyrophosphate,
Exactly. If you know this mechanism, your sequencing will be very easy.
pyrosequence.
Understand? Yeah. Not only pyrosequencing, everything. Okay. Like for example, when you add the DNTPs to the growing nucleotide chain, what are the things will release along with the pyrophosphate? Only pyrophosphate is going to release.
No, sir.
Then what are the things will going to release? See, in genetics, so many things it looks very simple, but actually it is difficult.
H plus ions also release, no, sir.
Yeah,
Ion
exactly.
torrent
Yeah.
sequencing.
Even water molecule also released. The my question is, how many water molecule you going to release?
One molecule.
Yeah, check it. Okay. These were the questions. Actually, it's about 10:40. I'm going to wind up. The question is, you write the equation of the polynucleotide chain. Sorry, how polynucleotide will there? What is the structure? Plus, when you're making it as a polynucleotide, how glycosidic bond will happen? How the phosphodiester bond will form? During the formation of phosphodiester bond, how many water molecule will release? During the formation of the glycosidic bond, how many water molecule will release? When this whole nucleotide will enter into the nucleotide chain, how many water molecule will going to release? Please work out this and please answer tomorrow. Okay, these are very important. If you want to know about the different sequencing technology, this is the basic thing. Why? Based on the release of H plus, there is ion torrent sequencing technology. The based on the release of pyrophosphate, there is a pyrosequencing 454 or pyrosequencing strategy. Based on the addition of nucleotide, there will be eliminance of there will be a release of there will be every time when you addition
Fluorescence.
fluorescence will release. If you instead of normal nucleotide, if you add fluorescence labeled nucleotide, there will be a fluorescence elimination. Based on this, Illumina will synthesize. Illumina and PacBio will come. Yes, Illumina, PacBio, whatever it may be, and Solid. This sequencing platform will come. That's why understanding the basic mechanism, how the the DNA is built and how the nucleotide synthesis will happen. This mechanism is very important that can be exploited in the sequencing technology. Now is coming. Then I hope you understood. Like I'm not told anything, but why I'm not discussing all the things, because if I discuss everything, each and everything in detail, I'll never go. It will take nearly. You know how many courses you will going to cover entire these things? How many credits you have taken in MSc? Total credits in MSc program. Remember how much credit you need to complete?
70.
Yeah, see 70 plus credits. See, if I explain everything, each and everything in detail, how much like see 70 credit in 1 hour, that too in 70 lectures, very difficult. That's why I'm focusing where you are not going to catch. These are the basic things. If you and explanatory, self-explanatory thing, this only you can get when you will read. Like when you read, revise, again you will read, you have to revise, again you have to read, you have to revise. There's the only one thing, only one way you have to get these things into your mind. Okay. Nobody will thought you like how exactly it will happen. And see the animated videos regarding the structure of the DNA, how actually, who is the person they are involved in the development of this structure? Can anyone tell?
Rosalind Franklin, sir.
Is the only one?
Wilkins Crick.
Wilkin, Wilkinson. What is photo 51? Can anyone know what is photo 51?
The X-ray crystallography image taken by the Rosalind Franklin.
Take Rosalind Franklin. She that's why regarded as dark lady of
Dark
the
lady.
genetics. But actually, Wilkinson and Watson and Crick, these were the three scientists. Actually, they are colleagues of them. They have some misunderstandings, so that they actually taken the information, but they're not given any credit for them. But before Nobel Award was announced, she was died due to cancer. But Nobel Award cannot be given to like cannot given be posthumously. That's why she is not awarded with the Nobel. But Wilkinson, Watson, and Crick, they are awarded with the Nobel Prize. And you can see the papers in 1941, 1942, 43. I think three significant papers you should remember with the journal name. Okay, that's the assignment. Okay. First, you write the chemistry in a single line. Then, what are the journals where this significant papers was published along with the years? Please write it. And while reading about this, you will get more information. Okay. Then you remember this chemical structure. And if you remember these scientists, it's more than enough. For ASRBNet, what are the information I presented in the slide is more than enough. Okay, no need to remember all the things which I have discussed. But for the SRF, it is must. For ASRBNet, it's not needed. Then, what is central dogma of genetics? Can anyone tell who proposed it?
Crick.
Crick, sir.
Yeah. When actually he proposed the structure of the DNA, in that paper only he mentioned about central dogma. Okay, it's not like in separate paper he not done. Well, he's submitting that paper only. He said that it will serve as a template and it will give as RNA and it will give for the protein. Okay, it is simple. I think many of you know that the information present in the DNA is transferred to the mRNA, then to the protein. Okay. Then this is called as transcription. Then this is called as translation. Then again, a separate studies. To study the transcript by different omics technologies called as transcriptomics. If you study the protein, it's called as proteomics. Again, it will the proteins will give state to metabolites. We studies the metabolites by using NGS, like advanced technology. It's called as metabolomics. Metabolomics. Then it is genome. Entire thing along with this, it is called structural genomics. These are all functional genomics. If you study the structure and organization of the gene, this is called as structural genomics. These things you should know. What is structural genomics? What is functional genomics? In functional genomics, what will come? If you study the transcript, then it is called as transcriptome. If you study the protein, it will be regarded as proteome. If you study the metabolome, if you study that is regarded as metabolomics. The entire
Sir,
thing it is called as functional genomics. Okay, what is
sir,
it? Yeah, yeah.
sir,
Tell me.
can we call this functional genomics as reverse genetics, sir?
Functional as reverse genetics.
Yes.
What is the relation between that?
Means in
Reverse
reverse
genetics is
genetics,
reverse genetics is a different thing. Okay, reverse genetics and forward genetics is the different thing. Okay,
because
what
in
is
reverse
reverse genetics?
genetics, sir, in reverse
There
genetics,
are
know the sequencer and we are attaching that sequence to a specific function.
no. I'm not discussing anything about that. Okay, that is a different. This is different study actually. It is nowhere it is related to this thing. Okay, it is the study. If you study the DNA, it is just you don't like you don't make a web among these things. I will explain why you not to make the web among these things. For example, DNA, RNA, protein. We study only the DNA. The entire thing is genomics. Okay, the entire thing is genomics. I will come to this in day after tomorrow lecture. For your interest, I'm telling. Genomics, entire thing is genomics. You study DNA, RNA, protein, everything is genomics. In the genomics, there are three parts. Can anyone tell what is the three part? Structural genomics, functional genomics, comparative genomics. Structural, functional, comparative. Okay. What is structural genomics? If you study how the genes are present in the genome and how it is organized. In day after tomorrow, I will going to deal with the genome organization in eukaryotes and prokaryotes so that you will get to know what is genome organization, what is paradox, what is C value, G value. Okay, those things I will come. If you study those things, like for example, size of the gene. If you take rice, it's for the TMB. It is a structural genomics. But if you study after it expression, when expression will happen at RNA level, protein level, metabolic level. We are studying the gene at its expression level. It is called as functional gene, functional genomics. Okay. Then we study, for example, you are taking a one variety and another variety, or for example, you are taking Arabidopsis and rice. We are comparing the genome of these two organisms by based on these information, structural genomics and functional genomic information, that is comparative genomics, like based on orthologous and homologous sequences. Understood what exactly these functional genomics, structural genomics? Okay, it is nowhere related to the reverse genetics and forward genetics. It's a different aspects. You should study in the different way. Understood?
Yes, sir.
Who, yeah, who asked you? You understood what is exactly these things?
Yes, sir, I
It's nowhere
understood,
less.
but I got doubt when I studied about reverse genetics, sir. It is mentioned that in reverse genetics, we will assign some function to the known DNA sequence.
Yeah, that can be done. That is reverse genetic tools. See, the phenotype to genotype. If you're studying the phenotype to genotype, it is called as forward genetics. If you have a gene sequence with you, you are assigning function to that genotype to phenotype. You are going, it's a reverse genetics. Means, for example, you take, we have, because when I was dealing with in nematode resistance, what I have, for example, if you know that this is a genome from 5 MB to 5.0 MB, you already know there is a gene present. But this gene is associated with nematode resistance, I don't know. What I can make, in what studies I can make that, I want to say that this gene is associated with the nematode resistance. What I can make, I already know the gene, where it is there in the genome, but I don't know what is the function of this gene. Either this gene is coding for nematode resistance or I don't know. How to do that?
You can develop the marker using that particular sequence and see whether this nematode resistant all plants
No,
has that.
no. That you already know the gene. If you know the gene, why we need a marker?
RNA interference.
See, I will come to it. Marker is used for when you don't know the gene. We go near to we map marker nearer to the gene, then we'll use it. I told that you know the information, you know the what is the gene itself. Okay. Then you don't know the function. We should assign some function. Then there is a tools called as reverse genetic tools. There are reverse genetic studies. Either you can use, can anyone tell any techniques?
Knockout.
Knockout, insertional mutagenesis,
RNA
insertional
interference.
mutation, RNA interference. Okay, then CRISPR-Cas9, and you can use
SD.
virus-induced gene silencing. Either you can use transposon tagging. There are several methods. Okay, these are called as reverse genetic tools, where we know the genotype but we don't know the function. Then we will use these type of tools. Then we will get to know, okay, this region of the genome may goes for the particular trait. This happens only when we know the complete information on the gene sequence, complete genome sequence. Okay, understood what I am going to say?
Yes, sir.
Then phenotype to genotype studies, what Mendel studied is completely forward genetic. We saw the phenotype, then we say, okay, some factor is governing the phenotype itself is a forward genetics. And he showed that marker identification and selecting, it is forward genetics or reverse genetics.
Forward only, sir.
Forward, sir.
Yeah,
Sorry, forward.
forward. Yeah, don't know, not sorry, but should know. Then anything? Tomorrow I will continue recombination. See, with this, and you have to read yourself transcription, translation, and these things. I will not go in detail as I already mentioned in the topic list. Then anybody have doubt regarding these things? Tomorrow I will deal with linkage and recombination. Actually, today I want to complete, but it has not happened. Linkage and recombination, how to create the maps, either genetic map, cytogenetic map, physical map. That I will going to cover tomorrow, like 3-point test cross, 2-point test cross, what is mapping functions, question be Holden. Tomorrow I will going to discuss in detail. After that, I will start molecular markers, mapping populations, QTL mapping, GWAS, and then I will move to MAS strategies.
Sir,
Then I will,
whether you also teach sequencing, sir? I'm asking because
yeah. When I start molecular breeding, exactly. See, what is SNPs? If you want to know SNPs without knowing sequencing strategies, how will you go to know SNPs? Single nucleotide polymorphisms. If you ask, sir, how can you identify single nucleotide polymers? Then if I don't tell anything about sequencing, then how can I say that how to identify SNPs? How can we detect SNPs? Can anyone tell? Venkat?
Um, mm, through high throughput sequencing methods, I think, sir.
Yeah, that only. Before, I think you read about this book, Marker Restricted Breeding by
Yes,
Singh
sir.
and Mary Singh. Yeah. In
Somewhat,
the
sir.
I am following the same every chapter. I am following in detail. Okay, each and every chapter, each and every topic, I am going systematically. Actually, this course is designed based on that book only. Okay? If you observe the topics present in the what are the topics I have given for the lecture series, it exactly covering the entire book up to 13th chapter of, which is very important for SRF actually. Yes,
Sir,
I can follow.
phenomics won't be asked as a paper.
Genomics, genomics.
Phenomics, sir. I'm asking. Phenomics.
Yeah, phenomics is very easy thing. One day I can finish that. No worries. One day you can finish the phenomics because there is only hyperspectral images, and I will show the virtual tour of the phenomics. I have that virtual tour video. Yes, the phenomics head is my member. I can ask him. Okay. Then anything from regarding this class? I know it is very. Today I'm very fast. Surface. Because now these things are you have to read. Now where you are already crossed the stage, because this is the basic things you should remember. Okay, the sex determination, sex linkage. Okay, which is the first trait. Okay, tomorrow anyhow I'm dealing with the linkage. That's why I not discussed sex linkage. Okay. Any doubts regarding these things? Vinay,
Not
Satya is very good. He already read. Pavitra, any doubts?
No, sir.
Sunandan,
It's okay, sir. It's a very nice class, sir. I recalled a lot of
thank
things.
you. Hey, thank you. Okay. Santoshini, any doubts? Abhishek,
No,
any
sir.
doubts?
No, sir.
Yeah, okay. Soumyajit, you joined late. Any doubt?
No, sir.
Yeah. Okay. Okay. Thank you. If no doubts means shall I end the class? Tomorrow we will discuss the linkage and recombination in detail, and that class only it will be start the basis for the molecular breeding. Okay.
purpose.
Okay. And I'll start. Okay, sorry. Before starting molecular breeding, I will complete. Forget it. I will complete the classical plant breeding. If you don't know classical plant breeding, it's very difficult to understand. I will start the lecture series of plant breeding. Sorry. Okay.
Purpose.
Thank you. And all. Bye.